this webinar is being recorded as a video, and no video or audio of attendees is being recorded. It's just myself and the host. And the speaker, Colin Forsythe, who's here with us tonight. Thank you very much, Colin, for coming. My name is Brad Nickerson, and I am a member and past president of the Belleisle Lake Watershed Association. And we are very grateful to Colin Forsythe for taking the time to prepare the webinar that he's about to present, and for sharing his deep knowledge of testing for cyanotoxins with us this evening. We have a really interesting presentation for you that Colin does. But before we get into that, I just want to mention that the Belleisle Lake Watershed covers 3,950 square kilometers, which is more than 5% of the area of the province of New Brunswick, and it's about 70% of the size of Prince Edward Island. So it's a pretty big watershed with a lot of streams, a lot of water bodies here in our watershed. It's a sub-watershed of the 55,000 square kilometer Wolastoq or Saint John River watershed. And Grand Lake, at 171 square kilometers, is the largest freshwater lake in the Maritimes. If you're not already a member of our association, we definitely would invite you to join. Memberships cost $10 per year and support our activities, including the organization and hosting of this webinar. Visit our website and click the blue Join Us button in the upper right corner if you'd like to join. And as I mentioned already, we welcome your questions. And the idea is to use the chat window that you're already, like I see a good number of you are already using, to ask your questions as you think of them. That's the best time to ask them. And then stay to the end of the presentation. Colin Forsythe will be more than happy to answer or attempt to answer any questions you might have. And due to the time limits, questions will be answered following the presentation in the order that you've typed them into the chat window. So I'm keeping a record of when they come up. And at the end, we will answer the questions. And if one is particularly general, that I might interrupt Colin. Hopefully, not too many times, Colin. But we will. If we run out of time at the end, we will send answers via email after the webinar to anyone who's asked them. If assuming we have your registration information, which we have an email for you. Okay. I see we already have one question, but I'll leave that for question number 1 on my log. Thank you very much, Scott, for asking that question. All right, so we're now going to have a quick poll, and I'm going to ask this. I'm going to stop screen sharing here and ask a question that hopefully everybody knows the answer to. And I'm going to have a quick poll just to give Colin an idea of the audience that's present at this point in time, what kind of knowledge they have of this topic. So this is a multiple choice question. You can only answer once. There's only one square you can answer. And if you just click the button that you think is the correct one for where do cyanotoxins occur, that'll give Colin a bit of an idea of what the audience knows and what he can focus on during the presentation. And I see we're up to how much percent? 60% of participants have clicked one of those buttons so far. And we're up to 76%, maybe a few more percent could answer what they think is cyanotoxin occurrence locations or location. I've only given you 5 answers. And it's good. We're getting some responses. We're up to 84%. Let's crack the 90%, and then I'll end the poll and everybody can have a look at it. Yeah, it only takes a second to think about it. Oh, we're at 92. Anybody else? Final chance. I'm going to end the poll. Everybody can see what the answers were. Hopefully you can see these results. Can you see them, Colin? I know no one else can speak,
I
but
can.
you can?
I can see the results, yeah.
Okay. So that tells you that it's kind of split between the last two.
It really is. Yeah, it's pretty close.
And there's, I guess you'll answer the question. I mean, I'm not exactly sure of the answer myself, but you'll answer it as we go along, or do you want to answer now?
Well, I can certainly answer it now, Brad.
Okay. Yeah, why don't we?
And the long and the short is that because cyanobacteria that produce those toxins can occur almost everywhere, and do you find them in marine systems, you find them in fresh water, you can even find some cyanobacteria growing in little ponds in forests or seeps and stuff like that. So where you have cyanobacteria, you can potentially have cyanotoxins. So almost everywhere, actually, they can actually turn up. But most people would probably recognize them from freshwater ecosystems because that's where we particularly here in New Brunswick seem to really speak about them.
Yeah. Okay, very good. So I'm going to mention a few words about Colin's background for those who haven't read it on our webinar invitation. And just a few words about Colin. Colin is currently serving as Executive Director of the Belleisle Watershed Coalition, beautiful area of the Wolastoq. For the past 3 years, he has been collaborating with watershed partners to investigate causes, conditions, and factors influencing the increase and formation of cyanobacteria blooms or mats and associated cyanotoxins observed in the Belleisle and Lower Saint John or Wolastoq River watersheds. Colin holds a Master of Science in Environment and Management from Royal Roads University in Victoria, British Columbia, where his research investigated microplastics in the Bay of Fundy. He also holds a Bachelor of Science degree in biology from the University of New Brunswick, focusing on aquatic science. He's a 2017 recipient of a Governor General's Academic Gold Medal for research on aquatic pollution in the Maritimes. And he has years of scientific, environmental, and project management experience in the nonprofit, government, and private sectors. So, Colin, I'll ask you to share your screen, and it's over to you.
Okay. Well, let's share that screen and see how the PowerPoint presentation works tonight. Okie dokie. Let's go play from start. There we go. Okay. I think we got it going now. All right. So I assume everybody can hear me at this point, Brad. So this is our little presentation on cyanotoxins, which has been an emerging water quality issue for the last probably 3-4 years at least in the Belleisle Watershed, and of course in the Saint John Wolastoq River as well. And I'm just going to draw you quickly to the photo there that was taken by one of our board members who has amazing drone skills. And he took that on the 5th of November, 2021. And that was a very large cyanobacteria bloom that occurred on the north side of Belleisle Bay, up around Hatfield Point. It actually stretched from Hatfield Point all the way down to the ferry landing, so several kilometers along the shoreline with very, as you can see, thick paint-like cyanobacteria bloom. That was my first major cyanobacteria bloom that I had seen, and it started me down the path of learning about cyanobacteria and the cyanotoxins they produce, which ultimately brings me here. So I'd like to thank, of course, our project funders. who provide the funding so that we can do these kinds of water quality projects. The New Brunswick Environmental Trust Fund. They support many great projects throughout New Brunswick for numerous watershed and other groups. And I don't think a lot of us could survive without them. Also, big thanks to Canada Summer Jobs Program that helps us hiring the students so they can get experience in the sciences as well. And of course, the seed program through Working NB that also allows us to hire some students for that. Without them, our projects would certainly not go forward. I would be remiss in not mentioning our project partners as well. Of course, the province of New Brunswick and the staff at ELG, particularly in the surface water division, have been incredibly helpful over the years with cyanobacteria and with our other water quality stuff. And I always look forward to chatting with them and see how we can help one another. I also want to put a big shout out to our watershed partners, Hammond River Angling Association. Sarah and her team over there have done an amazing job with the AlgaeTracker buoys I'll be speaking about a little later, and has been a thrill to work with, as has Roxanne and her team at ACAP Saint John, who provide the dip tip or rapid testing kits we use for the cyanotoxins. And we couldn't accomplish a bunch of the stuff we do without the help of these two amazing watershed partners. I can't thank them enough. So let's jump right into her. What is cyanobacteria? A lot of you probably know it as blue-green algae, but it is a type of bacteria, a microscopic bacteria that occurs in aquatic ecosystems, both freshwater and marine. Cyanobacteria is fascinating in that it originates about 3.5 billion years ago. It's one of the Earth's first life forms. And it is responsible for the production of our oxygen-nitrogen atmosphere. Prior to that, the planet had a very lovely methane atmosphere, one that we seem to be desperately wanting to restore through our actions, but hopefully we will be thwarted. These system, I have had people actually ask me if they can pour algicide in the river at their camps to get rid of it, and have someone horrified said, no, please really don't do that, and here's why. And they can be found in all of New Brunswick's lakes, rivers, wetlands, and marine ecosystems. They're always there. We just see them when we get the blooms. They really are an essential part of the aquatic ecosystem. They form the basis of the aquatic food chains in the river that support more charismatic species like Atlantic salmon, for instance, and eventually things like eagles and osprey, and even us. So life as we know it would not exist here without cyanobacteria way down there at the foundation. Now, the picture off to the side is interesting because that's a photo of some of the cyanobacteria strands from the bloom that occurred on November 5th, 2021. And I popped her in under my microscope and had a little look at it because I'd really never had a closer look at it before and wanted to see what it's fresh. It actually smells like freshly cut lawn. I can also tell you that when it goes off, it smells like really hot, rotten garbage. So it smells pretty bad, but this is the stuff that people are getting increasingly interested and concerned about. But it is really critical. So next we have to talk about the things that it produces. So it's really important to note that not all cyanobacteria blooms are going to be harmful. Some do produce toxins. Now, the ratio that's sort of been tossed around about that is 70% of blooms are not toxic and 30% are. And those 30% are the ones that we are generally concerned about. And I'm sure as we acquire more information and data, we'll be able to refine some of those numbers more. Certainly the majority of blooms I see in Belleisle are not necessarily toxic. Cyanobacteria blooms can produce a toxic byproducts, which we call the cyanotoxins. Cyanotoxins are basically a chemical toxin produced within the cyanobacteria cell. If you look to the photo there, off to your left, that's an even more magnified strand of cyanobacteria. That's not mine. That comes from a website online where I found it. Really, the photo really clearly shows it. And what happens, and I'll get into this in just a moment, because right now we know that there's about 84 cyanotoxins currently known. I can't list all of them for you. I have a slide that talks about a little, that identifies some of the more common ones. Basically what happens is when the cells on these strands die and they rupture, they release their cell contents. So if those contents happen to include a cyanotoxin, they'll release it into the water. As you can see by the first picture that we started with, you can get quite a lot of toxin fairly quickly. Some species are capable of releasing toxins into the water without cell death or rupture. And I don't have a full list of those either, unfortunately, but they are out there. Generally, what we see is toxins that are released after the death of the organism and the cell has ruptured. So there are a number of common cyanotoxins that we see in the surface waters around here. I've highlighted the ones that we have detected the most in our tests, partly because those are the ones we're keyed into looking for. Those would be anatoxin and microcystin, but there's, as you can see, quite a number of them. I don't think I will murder the Latin language this evening, but there's quite a number beyond that. But these ones are the ones, these 7 are the ones that we would expect to find more commonly in surface waters, and if we refine those, even more anatoxin and microcystin. Okay, cyanotoxins in Belleisle. This is another bloom in the photo there that came from one of our community members who was actually kayaking in it and took some photos and then wanted to ask us what it was. I believe that photo was taken in 2022, if memory serves, and fairly early in the year, I think. I know I have it somewhere with the date on it, but that green stretched all the way across the top of the bay. So the most commonly found toxin we see in Belleisle is microcystin. It can cause skin and eye and throat irritation if you were to handle it and get it on your skin or in your eyes, or you happen to be swimming and ingest it. If you ingest enough of it, you would get some more severe illness. I believe there's a slide upcoming that will mention that. None of which is fun. Anatoxin has also been found in Belleisle in benthic mats that occur in some shallow areas. The mats produce the toxin Cyanotoxin A. Cyanotoxin A is a neurotoxin, so it will affect your central nervous system. And memory serves, if you ingest enough of it, you will actually die from not being able to breathe, as it shuts down your respiratory and circular systems. But Cyanotoxin A is noteworthy, partly because of how severe the reaction to it is, and that it's the one that caused the unfortunate dog deaths that made the news in 2018 and 2019 in New Brunswick. And we're certainly very keen to test that and find out where it's occurring and how many of those mats are toxic. Those are pretty key. So we get different types of blooms in Belleisle. We get surface blooms. And they can look like the scum or foam or pea soup, or even little paint flecks or streaks in the water when they're sort of building up or on the decline. That photo is a shoreline, actually the mouth of a creek in the upper part of the bay. And it, as you can see, very pea soupish, and very, very much almost like a greeny scum on the water in part of the photo. It's often a blue-green color. Actually, if you see it dry on the rocks when it's fresh, it looks a lot like copper, weathered copper that gets that bluey green color. It's very, very distinct when you see it. It can also look red, brown, or different shades of green. I haven't seen it in really anything but the coppery color and this really bright green in Belleisle so far. Like I had mentioned earlier, fresh surface blooms smell like freshly mowed grass. It's really quite a pleasant smell if you love freshly mowed grass. And the older ones do indeed smell like rotting garbage, because I keep, I'm sure much to my mother's chagrin, samples in the back of the fridge, just in case I need them in the future. So, but yeah, surface blooms are the most common type of bloom we see in Belleisle Bay by a long shot. Like 99% of what we see for cyanobacteria blooms in the bay are these surface blooms. Generally, I've found that they appear after heavy rain, and when the water gets a little warmer. Kind of July through November area is when we see the majority of them. We had one bloom in 2021, the November 5th bloom that occurred in the fall, that I think was really a surprise to everybody, including myself. And then we had in 2022 8 confirmed blooms and 1 confirmed mat. Last year, we had 7 confirmed blooms, but we did not find or see any mats. So we do know that some of our blooms have and do contain the cyanotoxin microcystin, which is the skin irritation one, and the one that can give us. And I do have a little anecdotal story about that, and you can kind of see what it looks like when it's in a little sample jar. I'd gotten a call to visit one of the residents who had recently purchased cotton further down Nile and on the North Shore. And anyway, had giant hogweed. So not related to cyanobacteria at all. And of course they didn't. They had woodland angelica, because if you see giant hogweed, it looks like, really does look like some historic and is going to show up. And I said, no, you don't have giant hogweed. You have woodland angelica. Yes, it can cause some of the skin irritation. And as we were talking, they were starting to tell the story about how they had this green pollen that had shown up on the water around the dock, and they had gone swimming in this green pollen because their neighbor had told them that that's what it was, that it was green pollen and it was completely safe. You don't get a lot of pollen in August, just saying. And they had noticed that they had gotten quite sick afterwards that evening with headaches and some gastrointestinal irritation. And they were kind of curious what that was. And I told them, I said, you didn't have green pollen because pollen is yellow. That was a cyanobacteria bloom that had occurred a few weeks earlier. And it was one of the ones that we had found as mildly toxic, and they had gone swimming in it and gotten sick because they took the advice of their neighbor that it was safe. And that showed us that basically the symptoms that they describe are quite accurate. That we also kind of dodged a bullet that could have been a far bigger health concern, and that there was a definite need for us to do even more outreach to dispel some of the misinformation that was floating around about this. So that was really, really interesting. I'm super pleased no one was severely ill or worse, but it did show that that definitely we needed to do more outreach. And if you see, like, I have this little slogan, this little rhyme that I give the community that is sort of a very generalization. And I tell them about if they're looking at the water, and I say, look, if it's green, it's probably not clean. Don't swim in it. And again, a really big generalization there, but it seems to communicate to people that, you know, if you see something that looks a little funky in the water, maybe not dive in, you know, maybe give us a call and ask about it and come down and we can do a sample, which is what we do. And we since then, it's gone really, really well at getting people to do outreach and ask about things in the water that they see. So it's been really good from a community engagement point of view. Other types of blooms that we get in Belleisle are benthic mats. And benthic mats look like clumps of vegetation on the bottom of the river. They can be black, brown, or dark green. I haven't seen that. I've seen some gray stuff on the shore after it kind of lifts off and comes ashore and dries out. But usually what I see it as is, in the photo that's to the right there, a yellowy, greeny kind of beige color. And that is a cyanobacteria mat, and that mat was toxic. And that mat occurred on a popular trail up in Hatfield Point, where a lot of people do actually take their dogs through. So we were really quick to get in and to test that with one of the ACAP provided cyanobacteria kits. And it showed that, yes, it was toxic. So we definitely alerted the community, you know, just stay out of the water there, keep your dogs on a leash and out, and we'll keep monitoring it and updating you. And this bloom, and you'll see it a little later, was active for almost two months. And it really looked like, when I saw it, as a biologist, I can say it looked like the rotting flesh of an animal like roadkill that's been on the side of the road for a while. That's what it looks like. Smells like that too, actually. But it's not super pretty stuff, but it's the other kind of thing that we do see in Belleisle, and we found it in 1 or 2 locations in the watershed so far. Because it doesn't stick out quite as much, people don't seem to notice it. Although I knew our outreach was working, because this one was brought to our attention by a resident who called me on a Sunday afternoon and said, I think I found that cyanobacteria stuff you're looking for. And I said, well, send me over a picture. And, with the wonders of modern communication, I had it in seconds and was like, yeah, no, that's exactly what it looks to be. I'll come up and have a look. So, but and that showed that some of the increased outreach we did was starting to work, and people were starting to recognize what this stuff was and who they can reach out to. So how do these blooms occur? Like, what? They don't suddenly just materialize. It's not magic, as disappointing as that may be. But blooms and mats generally occur when the bay warms up, typically, as I've said before, early summer through autumn, that July, November kind of corridor. Generally, we see them after we have heavy rain events. And the things associated with those heavy rain events are nutrients, particularly nitrogen and phosphorus that we see in runoff that come from agricultural fertilizers, manure piles, septic systems that haven't been properly maintained, and clear cutting and shoreline development that wash into the bay, and they'll introduce nutrients, bacteria, increased turbidity, which makes the water look cloudy. That's why it gets that chocolate milk look. It's not supposed to look like that. That's an unnatural condition. So and it just shows that you get a lot of runoff, largely from human sources, anthropogenic sources. So when those heavy rains wash lots of nutrients into the bay, well, they are food for those bacteria. That's partly what they use, because they are plant-like, to produce food and grow and thrive. And when you couple that, and I call this a cyanobacteria triangle of nutrients, light, and heat. Now, there's increasing evidence that heat plays less of a role, and it's much more of a nutrient issue. But when you have those 3 things and they line up on the right, cyanobacteria can grow very quickly. They clump together and they form a surface bloom or a benthic mat. And of course, what we want to know is how many of those mats are toxic, and under what conditions. And that's where Sarah at HRA and her wonderful technology will come into play, which we'll talk about. So this is a little map that I did up. I apologize that the 2023 blooms aren't on there yet, but they would look pretty much in the same locations that we're seeing on this map. These are the blooms that we've seen since 2017. I have records of one bloom that occurred in Urquhart/Browns Cove back in July, August of 2017. And that seems to be the first one recorded or on record. After that, we don't see anything until the purple one that's way up at the top there. That's hard to see because it's kind of buried behind a couple of blue balloons in November of 2021. And that was a large bloom. And then in 2022, they really start to take off. And for the eagle-eyed observer, they'll notice that a lot of those are occurring on the North Shore in the upper part of the bay above Earl's Wharf and Long Point, where that ferry crossing is. but that other parts of the bay are susceptible to them based on wind direction, current, and tide. Can move that stuff around very quickly. Because I've had people call me about cyanobacteria, and 20 minutes later, when it gets down the watershed to wherever they are, it's gone. It's moved out with the current or the wind, and it has disappeared. And usually I just say, if it pops back, let me know. I don't mind coming back. And that's it moves very readily. A couple of years ago, with the data that the watershed had accumulated from nutrient sampling from the water quality monitoring program, I generated this rough map of the different types of trophic states in the bay. So this highlights nutrient concentration in the bay of phosphorus and nitrogen. And I'll explain the trophic states because they have fun science words, which are fancy ways of saying things. So at the top of the bay, where it's bright red in Hatfield Point, that's what we would call in nutrient speak eutrophic. And really all we're saying there is that we have a high amount of nutrient in the water. And indeed, it's up in like the 0.3s, which is a little high, particularly in the summer. Again, most of the water quality data comes from the summer, so it's difficult for me to say what that condition is in the winter, although I suspect it decreases a little bit. I'm sure it doesn't decrease a lot, but we see higher concentrations of phosphorus and nitrogen in the upper bay. As we start to move down it, and I know I have to edit this map to put a little more yellow in, we get this kind of boundary zone that can come back as mesotrophic, which means it has a medium amount of nutrients, but it's generally healthy. And eutrophic, which says it has a high amount of nutrients in the water at certain times, particularly after heavy rain events when we've had lots of runoff. Some of the recent hurricanes have certainly done that. 200 millimeters of rain we got a few weeks back, that would certainly have added a ton of nutrients. I was actually tempted to go out and see if I could sample and check on nutrient levels with that. I didn't, because generally during the winter I'm mostly alone, and I try not to do field work on my own, because that's when all the bad stuff happens. And then of course, for the rest of the map, the green, that is mesotrophic. That's showing that generally what we have here is a system that has a moderate amount of nitrogen and phosphorus nutrients in it. It's healthy. It's going to support lots of life. It's safe to swim and play in. Don't panic. Now, what's really interesting is that if we could all pop into our TARDIS and go back about 500 or 600 years and take a bunch of water samples all through the watershed here, I suspect that we would find a word that's not on there called oligotrophic, which indicates that these ecosystems were nutrient poor in the past. And that's because there would have been a ton of forest, no septic systems, no summer cottages, no agriculture, no forestry. It would have just been whatever the First Nations were doing throughout the watershed at that time, which was probably subsistence living. And it would have been a very kind of different nutrient system in the past. The arrival of Europeans, who brought with them the Industrial Revolution and required the use of the land and the forests and the minerals and stuff in it to drive the Industrial Revolution, and indeed their economies and lifestyles, altered this system very likely forever, or at least until we decide that we're going to change how we do things. So it's an interesting map. It does need an update now, and hopefully I'll get some spare time eventually to do it, but it gives you an idea. And as you can see from the previous map, in the areas where we're seeing higher concentrations of nutrients, we're seeing more blooms, I guess is there. And in those same areas, of course, we're seeing more of the cyanotoxin. All right. Now, these are a number of our monitoring sites. Again, my apologies if they're a little difficult to see around the bay. Generally, we sample 3 sites that we are known for cyanobacteria and mats regularly. And they are the wharf site, the trail site where the mat occurs, and the ferry site. And as
Colin, sorry to interrupt. Could you
Yep.
maybe use your mouse to point out those sites as you
Sure.
say their names? Yeah, thank you.
So that is our wharf site at the Hatfield Point Wharf. Also our water quality #1 site. Oh, where'd my mouse go? #2 is here, not far from that, on a little trail that goes around the shoreline there. And then #3, the wharf site, is right down here at the ferry landing at Earl Cove for the Belleisle ferry. And as my summer students pointed out one day, and you'll see this in one of my photos, it gives us a lovely acronym of WTF. So, and once they pointed that out, I must say I have to laugh all the time. So when I see it, but those are the ones that regularly sample. I do sample the other ones when blooms are present, or when an individual in those areas would highlight to us that there is a bloom occurring, and we will pop down, jump into the old Batmobile and take off in a cloud of dust to get those samples, and hand out some cyanobacteria literature and stuff that is kindly provided by both the Department of Environment, and we have some from ACAP too. But those are the sites that we sample. You'll notice, yes, there is kind of a gap that goes down into Kingston Creek because we see practically no cyanobacteria there, but we'll sample there as needed. And now I want to get into some of the other tools that we have started to use for cyanobacteria. We monitor for cyanobacteria blooms not only through observation, as I've mentioned. All the staff are trained to kind of identify cyanobacteria blooms, and our outreach helps our communities do that. But this past year, HRAA, Hammond River Angling Association, offered us an AlgaeTracker sensor buoy. And man, I gotta say, this thing is fantastic. As a big science geek, I love this kind of stuff, but it is shown how it has been a valuable tool in learning about cyanobacteria. This sensor buoy, which we use when we set up around Hatfield Point, provides real-time data on a number of different water quality parameters that would be associated with bloom formation, including water quality. On water quality, including temperature, phycocyan, and if you're wondering what phycocyan is a fancy way of saying blue pigment that occurs inside the cells of the cyanobacteria, hence the blue-green algae component of it, and that's where it comes from, is the cyan in phycocyan is the blue pigment. And of course, the other one, which we're all very familiar with, if you've ever looked at a leaf or piece of grass, chlorophyll A in this case, that occurs because they are plant-like. So it registers both of those. It also registers things like turbidity, which is the cloudiness of the water, which is very handy, air temperature. So it gives us a number of different measurements in real time, tracks them, graphs them, produces tons of data, super fantastic. When high levels of phycocyan occur, for instance, 150 relative fluorescence units, or RFUs, and chlorophyll, when we see them above 200 RFUs are detected, we know now that a bloom is very likely intimate in the watershed, and that's when we will start to look and test and move on things so that we can catch that bloom, because we want to catch that bloom through its entire sine curve, right, or bell curve, as it's increasing, when it sort of crescendos and is at the peak, and as it descends, and see and track that, and track how it produces cyanobacteria through that curve, so we know when to look for it. And these are some of the graphs that it produces and shows it. And yes, where you see the great big spikes is where we get blooms. The really, really big one is just in and around the 27th of September last fall. I was actually out doing water quality with some of the Hammond River staff, and we came across this pretty massive cyanobacteria bloom in the top of the bay, and took a sample, and poof, there you go. It was definitely present, and as you can see, it turns real quickly from very low readings to quite high quite fast. On average for chlorophyll A, we found 119 RFUs in the bay during the sampling period. Our minimum was 40. It's not going to be zero, because there's always going to be some kind of byproduct of photosynthesis, some kind of chlorophyll in an aquatic ecosystem, or at least there should be, particularly on surface ones here, and a max of almost 3,000, which was quite interesting. And I want you to keep an eye on these graphs as well, and see where all the spikes are on them. This is the phycocyan one, nicely graphed in blue. And again, you're seeing the spikes in phycocyan. If I draw your attention to that one on the 27th-ish area of September, a great big huge spike. Now, because phycocyan is related to cyanobacteria blooms, you can have a minimum of 0 of it detected in the water. Part of that has to do with the sensitivity of the instruments that you're using. I'd say that the AlgaeTracker is fairly sensitive, so and we averaged over our season 31.8 RFUs of phycocyan, and a max of just over 1,000 phycocyan with that large bloom in September. But it was very interesting to watch it and track it. Water temperature, the spikes in water temperature generally occur a little bit before the bloom. It's interesting because if you line up that spike in and around the 27th, it occurs just after a higher water temperature. But generally what we see is water temperatures that were between 15 and yes, we did have one day where the surface water temperature was 30 degrees back on a super hot day in July. But on average, water temperatures in the surface waters of the bay are 21.9 degrees Celsius, a comfy temperature for swimming in the summer. But that's seeing, and as you see, they decline as we move through the summer season and into fall. And if we had them for several years and could kind of pan out a bit, we just see a very lovely sine wave that kept going with water temperatures going up and down and up and down and up and down and slowly creeping up because of the often heard climate change altering the temperatures of our surface waters. Now, turbidity, which is the cloudiness one that I had mentioned, and this comes from our AlgaeTracker. As you can see, we can quickly point out that that September 27th area great big huge spike in turbidity that's associated with increase in phycocyan and increase in chlorophyll. So we know when we start to see things like cloudier brown water, we want to be checking. This all links up really, really nicely. I actually have a page where I've lined them all up so that you can just look straight down and see and line everything up and look at all the different spikes. We had an average NTU of 34, and a minimum of 3 and a maximum of 400. 400 is really high, right? Generally for turbidity, you want it to be kind of between 0 and 10, and then after it passes 10 and starts to go up, it's increasingly turbid and not so good. It would be like, for us, it would be like being in a sandstorm or a blizzard. And you certainly wouldn't want to breathe the frozen snow particles or sand particles through your lungs. Well, fish have to breathe that dirt through their gills when they get the high turbidity in the water. So I always feel bad for fish when I see that. Now, on to our cyanobacteria testing, or the cyanotoxin testing more specifically. We use rapid test kits. They come from Eurofins Abraxas. They were kindly provided by ACAP Saint John through a partnership project where they're doing some larger research on cyanobacteria. So we're very pleased to be a part of that and to have the kits to do the testing. Due to unique properties of the toxins, we actually have 2 different types of test kits. One tests for microcystin, and the other one tests for anatoxin A. Each of the kits supply all the necessary bits and reagents to do a respective analysis. You can do 24 analysis with each kit, 24 or 20 analysis with each kit. And they are what we would call a semi-quantitative dip strip that screens for a total of microcystin or a total of anatoxin. For microcystin, it's between 0 and 10 parts per billion. And for anatoxin, it's between 0 and 2.5 parts per billion. To provide a quick indication if the toxin is present in the sample. So when we have a bloom, we will go out and we will sample all of that stuff. You can take the kit with you and basically do it in the back of your car if you really want to. Because most of our sites are pretty close to our office, I usually just pop them into the cooler with some ice packs, rush and grab them and get them back, and then sit down with the staff and do the tests. And starting actually last year, we also took a new path on cyanotoxin testing by also collecting water samples that go to RPC in Fredericton for a more quantitative analysis. And that was really, really revealing in and of itself when I get to the results here. So this is what the tests look like after they're done. This is a test that we did for a bloom on the 12th of September this past year. We did anatoxin at our Wharf Trail and Ferry site and microcystin at our Wharf Trail and Ferry site. We do that every time we go out, and I try to sample once every 2 weeks, particularly once every 2 weeks once we hit that July to November corridor. There is a period in August where it sort of drops off if it's really dry and we don't have a lot of rain input. And you can kind of keep a different kind of eye on it there. I'm sure you're probably all wondering, all of these tests came back as negative. So this is where it's semi-quantitative. So we get 2 colored bars on the strip.
Could you show where sorry, Colin, could you show where those are on the strip there?
Oh yeah, so it's these 2 strips right here. that run down, and one of them is the control strip, and one of them is the test indicator.
Mhm.
And basically, once they've gone through the whole reagent process, you'll get these two bars, and then you have to hold a sheet up with those bars, and the sheet shows, oh, this test is negative, this test has a concentration of 5 parts per billion, this test has a concentration of 2 parts per billion. So you kind of have to eyeball it and guess it a bit, and that's where it's semi-quantitative, because it depends on the tester looking at it and having a lot of experience to say, yeah, I think it's here, or yeah, I think it's there. So semi-quantitative, but what it does say is, yes, toxins are present, or no, they are not. And that's sort of how we treat them. So we don't put up in our posts concentrations generally. We had initially, but we've kind of shied away from doing that because we can't really guarantee that that's the exact concentration. So yes, it is toxic, or no, it is not. Because of these test strips all came back negative. Basically, the equal color intensity on them, and I know you're looking at some of them and saying, well, they're not equal in intensity. When they're there in person and not through camera and stuff, they look much more. But yeah, that's the kind of challenge you have. Then they would be below the detection limits of the test, and therefore negative concentration of microcystin or anatoxin A. The test strips have made a really big difference and is one of the tools, along with the water quality testing and the cyanobacteria, AlgaeTracker buoy that we're using to flesh this out. So here's some results from our testing. This is the results of our microcystin testing that we did with the November 5th bloom that we saw at the beginning of the slideshow. This one came back as less than 0.5 concentration of microcystin across all the sites that we tested. So at the time that we tested that on the 5th of November, very, very low, almost undetectable amounts of microcystin, but some that was present there at that time. Those were all the test strips, unfortunately, that year that I had left. And as a result, I wasn't able to do subsequent testing on that bloom after that. I certainly would have tested it probably every couple of days just to see, does that concentration increase as the bloom kind of dissipates? Is that concentration of cyanotoxin present after the bloom is gone? Does it remain in the water for a period of time? One of many questions. This again is another photo of that said bloom that starts to show more of its size. We actually think some of the blooms form on the south side of the bay and then blow over because of the southwest winds to the north shore rather than necessarily forming there. The upper part of the bay has a lot of development, agriculture, forestry. At least half of the watershed drains into the upper part of the bay through a number of different creeks and brooks. So it gets a lot of nutrient loading up there, which probably helps to make it the cyanobacteria capital of Belleisle. Also, it's more shallow up there, so the water tends to be a little warmer and more easily disturbed. So have a slightly higher turbidity during some events. These are the results from 2022. The number of blooms that we observed across our different sites and between July and basically the end of October. 8 blooms were observed between July and October. Most of the blooms occur in the upper bay. So as you read the names across the graph, they start at the upper part of the bay and they end at the lower part down around Carrs and Anderson's Point. So we see quite a bit up in Hatfield Point, Kierstedville, and Earl Cove, where the ferry is. And then as we go below that or over to the side of Long Wharf and move down Long Wharf and Herbs Cove, and then jump back to Jenkins Cove and Carrs, we see quite a big decline. They did get a big bloom in October after quite a heavy rain event that year. 2022 was the year that we saw the benthic mat at the trail up at Hatfield Point, and that benthic mat appeared in August and stayed with us to October. And these are some of the concentrations, because of course we did the cyanotoxin testing with the kits. This was before we were sending water samples away and before we had our buoy. So we had only the test kits to do that. And we found that 2 of the blooms tested positive. One of the blooms in July, which seemed to be in and around 1 part per billion, is our best estimate. And a large one that we saw in October, that one was in and around Thanksgiving. That, depending on where you were in the watershed, registered between 5 and 10. So the sites at Long Point, Herbs Cove, Jenkins Cove, and Carrs were all down around 5, and as were Hatfield Point, Kierstedville, and oddly enough, Earls Cove tested way up at 10. So certainly we seem to see more blooms in the fall than we do during the actual summer, but we can certainly get them through all points. There's sort of like a peak, and then it kind of drops off in August, and it picks back up in September, which is fine in the fall, because generally people and their pets aren't in the water as much then. This is our anatoxin A in cyanobacteria mat that we tested, and we started testing that in August, and it tested for that kit it was pretty strongly indicated at 2.5 parts per billion, and it kind of really sat that way until the water started to cool in October. At which point it dropped off to 1 and then kind of disappeared in November, and has not been seen since. It need not come back, actually. So it was interesting that anatoxin A was detected from the same mat for 2 months. So this clearly was continuing to grow and die and produce anatoxin A for a long period of time in the water. But once the mat disappeared, it did indeed, the toxin did indeed disappear as well. And we found that really interesting for just how long the mat could exist and continue to produce cyanotoxin. Now, here are our results from last year. Definitely world exclusive. People have not seen these before, other than me and select individuals. So this year we had almost the same number of blooms as we had in 2023, and the fact that we've had back-to-back years with about the same numbers of blooms makes me start to wonder if we weren't seeing some of these blooms earlier because we didn't know what we were looking for, and they were just being missed because people just thought they were algae, which is possible. Again, speculation, but possible. But now that we're looking for them, this seems to be what we've turned up the past couple of years, is in around 8 and 7 between July and October. Again, what you'll notice is that the blooms really occur in the upper half, northern side of the bay. Throughout our surveys, I did not see anything at Long Point or Herb Cove or Jenkins or Carrs. it seemed to be reasonably devoid of any kind of large blooms that showed up, and nobody reported any of the blooms to me this year, and we didn't see them on our surveys. There were no benthic mats observed, and that's really interesting in and of itself, because this year we had a lot of cloudy weather. We had a lot of rain, like I like to say, biblical amounts of rain. And water levels in the bay, like many other parts of the river, were really high this year. And I'm curious if that combination of high water, cooler water temperatures, and cloudiness was breaking that cyanobacteria triangle I talked about. Because certainly there'd have been a ton of nutrients, but with all the water running off. But if that was breaking that triangle and placing us in a position where the mat could not form because it wasn't meeting all of its life requirements to do so. And I definitely waded in that area with my chest waders and had quite a good look for it. I kept an eye out for it because I knew what I was looking for, and it just never showed up. Shoot. So, but we did again do the cyanotoxin testing, and only 2 of the blooms tested positive. Both of them are in Hatfield Point. One, I'll put the little there. It's the first one, and it tested very, very weakly at about 1 part per billion, and that was in July, right at the beginning of July. And then the big red bar is the one that occurred on September 27th, and it was 50 parts per billion. And that was the one, it's interesting because the test kit that we did, the rapid test for that one that I did from the sample, it did show that the bloom was toxic. And it certainly maxed that test out. It was very strongly positive. But seeing as that one kind of stops at 10 parts per billion, we're several orders beyond that here at 50. And that was done by RPC. That was the water quality analysis that we sent off to them, the lab, the sample that we got, and it came back at 50. That's the highest amount of toxin that we've recorded in the bay so far. And again, that raises the question, you know, we definitely should be doing test strips, but in combination with the test strips, certainly sending water samples off when we can for analysis to get a more accurate picture of what the toxins are and the concentrations of the toxins in the water when we are having a bloom that is occurring. We did collect some anatoxin A samples to them. They're trying to develop some testing for that, so hopefully in the future that will become available as too. But yeah, it was a quarter year from toxin wise. Not quite as many blooms, and of course anatoxin A, we got nothing. Zip, zero, zilch. There was no anatoxin A toxin determined or observed or tested for. Well, we tested for it, but we never got any results on it in the bay. And this is interesting, and when I get to the conclusion slides, which are shortly coming up, mat formation this year, which would seem to inhibit anatoxin A. So the really important questions out of this cyanotoxin testing, excuse me, is what have we learned from it, right? Because it's great to put all spiffy graphs and maps up, but that doesn't mean anything if we haven't learned anything. And here's the little rundown. Blooms and mats generally occur in the upper part of the bay between July and November. I know, spoilers, right? No part of the bay is immune from cyanotoxins or blooms. We do see them in other parts of the bay. Some parts of the bay, it's rarer than others. Large blooms seem to occur in autumn, but we can have larger blooms at other times of the year, as indicated by some of the photos. But generally, it seems to be September through November when we start to see really large blooms. Weather patterns and wind and current and tide really determine the distribution of blooms in the bay. In the summer, we get very predominant southwest winds, so that's going to blow that to the upper north portion of the bay. Bloom and mat formation is strongly linked to nutrient levels in the bay. And nutrient levels in the bay have continued to increase consistently for the past 3 to 4 years. Our water quality information has shown that, and particularly increases in the upper part of the bay at Hatfield Point and Kiersteadville and down to Earl Cove. Consistently, we're seeing increases in both nitrogen and phosphorus. I have a little water quality graph in a different presentation that shows those changes in nutrients. Temperature seems to be less of a factor because in the November one that we had in 2021, we really thought, I had been told about cyanobacteria and was keeping a lookout for it that summer. And I figured at the time, oh, it's November now, we're probably good, not likely to there. Wrong. Wrong, because we must remember that these cyanobacteria evolved to live in this environment. They could well create blooms under the ice in the winter, and we wouldn't see them, right? Because we're not looking for them then, and they would be hard to detect under what ice we did get on the river this year. So there is that, but they do seem to occur more frequently during the warmer parts of the year. Certainly the warmer parts of the year, it's more conducive to grow things. I definitely would not want to be a cyanobacteria trying to grow in the river in January, but to each your own. What else have we learned? Not all blooms are toxic. In fact, the majority of our blooms, I'd say probably about 80% of the blooms that we see are not registering as toxic as we test them through their formation as they peak and as they fall off. 80% of them are not coming back as toxic, but that still leaves the 20% that are. And the 20% that are seem to be largely microcystin, right? Low levels for the most part of microcystin is what we found so far. I'm hoping that additional water quality testing in the future will give us a more accurate picture of the concentration of microcystin toxin in our blooms. In Belleisle, Cyanotoxin has only been detected in 2022 at Hatfield Point and only been associated with benthic mats. I have never detected Cyanotoxin A in any of the surface blooms. And in consultation with some of the other watershed groups, that seems to be some agreement that they're not seeing that as well. Maybe they have since found, and I haven't spoken to them recently about it, but that's generally what we've seen in Belleisle is if you have a surface bloom, you got microcystin, but you don't likely have Cyanotoxin at all. That only seems to occur with mats. Really very clear to me now is a combination approach of tools like the AlgaeTracker buoy, the rapid tests, and lab testing is really helping us gain information and fill those puzzle pieces in on cyanobacteria and cyanotoxins in the watershed. So certainly we need to do more research to help understand the what, why, and how around cyanotoxin and blooms and mats, like some more of what is causing it, what conditions are really ripe for that. Why are those conditions better than other ones? No. How are these things functioning in our ecosystem? So, and again, more public education is needed to help people understand the issues, challenges, and adapt with these changing conditions. I can tell you, if you want to reduce cyanobacteria blooms in your watersheds, you need to reduce the nutrient loading that's going in there, because boy, we're going full steam with nutrient loading on stuff, and we don't have to do it. We have on the ground solutions that we can do now, like riparian planting or planting along stream banks and shorelines to help put those buffers back. We could certainly adopt kinder, nicer forestry practices that would help to reduce runoff there. More work to fix and maintain septic systems, and certainly more education for people that own cottages and homes along the water, to help them be better stewards of the land and water around them would all help. These are all some of the things that you can do to help reduce it here in Belleisle Bay or anywhere in the river or lake. This is helpful. It's really a question of, what kind of ecosystem do you want in the future? Do you want one where there's cyanobacteria alerts in the summer and you have to tell your kids or grandkids, maybe we can't swim for the next week? Or do you want one where the water's really crystal blue and these things are fairly rare in occurrence, and you don't kind of have to worry, right? It's really up to us, because this is an anthropogenic problem. This is a problem that we caused by the way that we live, and as we learn and modify that, we can become better stewards of the environment and avoid some of the worst that could occur. So anyway, if you're looking for additional information on cyanobacteria, I highly recommend Environment and Local Government's website. They've got lots of great information there, with a very, very nice public orientation to it. You can also report sightings to the Belleisle Watershed, as you can to Environment and Local Government, whichever regional office you happen to be in. If you're in Belleisle, that's Region 4 out of Saint John. I'll tell you, ACAP also has some really fantastic information on cyanobacteria. And other than that, I will thank everybody for enduring this last torturous hour of cyanotoxin. You have made it to the end, and I will now attempt to answer all of your questions.
Okay.
But yeah, thank you. Thank you, everyone.
Thank you very much, Colin. That's an excellent overview. We've had one comment already that says, very interesting, and I've learned a lot. Look forward to watching the end part I miss on the recording. So this recording will be edited and posted on the Gem-Site Grand Lake Watershed Association YouTube channel. So stay tuned for that. Everybody that registered for the webinar will actually get an email saying when that YouTube video is ready.
Mm-hmm.
Okay, we have a number of very interesting questions. Scott Oikel and another gentleman, Charlie Taylor, asked, is there any future testing to be done in the Wolastoq Lake, or any testing for cyanotoxins in general in the Wolastoq Lake? Do you know the answer to that?
I cannot answer that question. There is a Wolastoq Watershed Association that would handle that. Obviously, Belleisle does not test there.
No, the Gem-Site Grand Lake Watershed does not test there either, certainly at this point in time.
But
so we know that, yeah.
I can tell you the Wolastoq Watershed, they have a long-running water quality monitoring program that goes back, I think, like 20 years.
Yeah.
But I can't tell you specifically whether they are testing for cyanotoxins or cyanobacteria at this time. So I would reach out to, I don't know if she's still operating as their executive director, but she certainly had in the past, was Sean Dalton, and I can't think of any contact information
Yeah,
off the hand,
I
but
know, I'm pretty sure Sean Dalton has stepped down as executive director of the
okay.
Washington
Yeah, I wondered,
Watershed.
I had kind of heard that maybe that was
Yeah,
the case. And
and I do have a contact that I might be able to dig out. So what we'll do for Scott and Charlie is we'll get that question posed to this person that we've communicated with from the Washotamowick Watershed Association and get back to you on that one. Okay, so Brad Crawford asks, is it easy to distinguish between cyanobacteria bloom and a pollen outbreak on the water?
yes, it is. It
Okay,
is super easy. It is a color test, and is it yellow? Then it's pollen. Is it green? Then it's cyanobacteria or algae. It's
yeah,
that simple.
yellow.
It is either yellow or green. Like
Okay,
if it's yellow, you've got pollen. And pollen you're going to see in large quantities in the spring and early part of the summer, generally before you see cyanobacteria. So in April, May, June, if you see a lot of yellow on the shoreline, kind of a yellowy scum, pale yellowy scum, or yellow like dust floating on the water, pollen, pollen, pollen.
okay,
If it's in kind of July through November and it is blood green, like pea soup, that is definitely cyanobacteria,
yeah,
right?
okay.
I'll bring
That's
you
a
back to
pretty,
that generalization.
yeah,
Yeah, if it's green, it ain't clean. It also not pollen. Pollen definitely appears as yellow.
yeah,
And if you happen to be a huge nature and science geek like myself, then invest in a basic microscope. And if you scoop that pollen up out of the water and put it on a little slide and magnify it, you can actually, there are guides that will let you identify what species of tree or plant that came from,
okay,
because pollen have all kinds of absolutely extraordinary shapes under the microscope. Trust me, you will not go wrong if you try that.
okay.
Don't, yeah, I don't know if you should collect cyanobacteria samples
No,
and do
I
it,
wouldn't.
but certainly pollen, you're a lot safer.
Okay, right. So that's a pretty clear answer. Thanks a lot for that, Colin. And we've got other questions.
Mhm.
Leslie asked us, I assume at some point Colin will address the rate of increase of the blooms, say over the last 50 to 100 years, as related to the causes. But I know you covered 3 years, but I don't think
That is an
within that
interesting
length of
question.
time.
No, although some days I feel like I've been around for 100 years. And if I have, then I look amazing. But there's just not data that goes back more than a few years to do it. If I were to graph that for you, it would go whoop like that. It just kind of, there is a bloom report in 2017. There seems to be nothing in 2018 and 2019 or 2020. There is the 2021 one in the fall, and then it kind of drops off. And then in 2022, it just sort of takes off there. I'd love to look at, I would love to see nutrient data from the river or even Belleisle that would go back as far back as those records go. And you could actually graph the changes in nitrogen and phosphorus inputs. And I suspect that graphing those would parallel increases potentially in cyanobacteria over time. But it would be a guess because nobody was looking for this stuff just a few years ago. I mean,
Right.
if you had asked me probably about 10 years ago what about cyanobacterial blooms, I would not be giving this presentation. Um,
Yeah.
I would have known what cyanobacteria was thanks to my biology and science background, but beyond that, it would not have been on my radar or a concern as a water quality issue a decade ago.
Yeah. Okay.
Um,
Fair
yeah,
enough. So it wasn't a problem, at least
no, it didn't show
in
up.
the Wolastoq, at least for that, that
Yeah,
long ago.
and I mean, 100 years ago, on the lower parts of the Wolastoq, it was much more developed than it is now. There's actually more forest there now than there was in the past.
Okay.
So they may well have gotten blooms, right?
Mhm.
And just, it would never have registered with them.
Yeah. Okay. We have another question from Charlie. Charlie Taylor asks, are test kits readily available to the general public?
Well, Charlie, if you do want to order one from Eurofins Abraxas, I believe you can do that. The kits cost, if memory serves, about $1,000.
Yeah.
And gives you 20 to 20, is it 20 tests that that you can do. I mean, groups like ours, I will come out and collect a sample and test it for you with our kit free of charge, which saves you, and we keep you in the loop in it. Like, it is really important to me to engage with our community and make sure that they're informed with as much facts-based information as I can get to them, because community members who are informed with information can make good rational decisions. When there's a bit of unknown around it, people are naturally a little uncomfortable and afraid of it. That's human nature, but once you start to flesh it out, that sort of it. So you go to Eurofins and Abraxas, I suspect likely you can get a kit, but they are expensive. I haven't heard of general members of the public purchasing the kits either.
No.
I've only seen them kind of distributed in through the watershed groups. That's an excellent question. Colin Forsythe, private citizen, may have to see if he can buy a test kit.
Yeah. My experience, I actually used a test kit or was involved in a demonstration of one, and it was non-trivial in the test that I was involved with. It's not just a 5-minute thing. You just pop a strip in and look at it. There's a number of steps. I think it took us about 30 minutes to do one test when we did it.
Yeah, that's when it takes us to do 6 tests. It takes us about an hour once we've got the motion down and know what we're doing.
Yeah,
So
and you need to keep the sample cool, as I understand it, before you do the test.
yeah, we keep them in the fridge. Yeah, they have a shelf life and a temperature range.
Right.
I generally keep them in the fridge and keep them cool. I think their shelf life is about a year.
Yeah.
Yeah,
Okay.
on them, but they do indeed expire eventually.
Okay, we have a question from Pam Fowler. She asks, is monitoring being done on Grand Lake? So I don't,
Well,
that's a good question for me to answer. I guess I know a little bit about it. I wasn't doing it myself. So there's no, at this point in time, Pam, there's no actual scheduled monitoring like Belleisle Watershed is doing. But on sightings, we will use our test kits that ACAP Saint John has kindly provided and actually go collect a sample or take a sample from somebody that's provided one and actually run it through the test. But this is for people within the watershed group that have been trained and practiced this a bit before and are familiar with it. Pam noticed that there was a picture on our invitation that had a Waterboro wharf bloom, and that was provided by just people that live in the area, as you've mentioned already for the Belleisle. Noticed this, took a picture, sent it to us. Is this a bloom? Is it toxic? So we followed that up, I think, that particular case with a visit. And the one on Fanjoy Point was also somebody that lived there, took the picture the day before, huge green bloom. Day after, it's completely gone. So you need to be ready to sample and test as soon as they appear. As you mentioned before, they can disappear quickly. So it's quite a process. There needs to be more volunteers and also more, I guess, watershed members that are trained in this process, so that and summer students, as you say, like in biology and whatnot, to help out.
Yeah, all the staff are trained
Yeah,
on the test kits each year.
yeah,
I think it's good education and training
right.
for them to have, and they all get trained. And if any member of the public did get a test kit and wanted to run through it, I would certainly be happy to sit down with them
Wow, that's a
wherever
great
they
offer.
are in southern New Brunswick and run through a sample with them as well. So the more people that have knowledge and are enabled, the better it is for everyone.
Yeah, and it's good if someone does buy a kit privately, it's good to keep the watersheds informed, because we collect data for the whole watershed, or try to, a portion
Yeah,
of the watershed, and have a historical record of what happened. Like your overview of what happened in 21, 22, and 23 was great. It's clearly showing an increase in the blooms in that time period. Okay,
yeah.
we have other questions. What is the communication process for real-time monitoring with the algae trackers? Uh, I know for a fact that that's part of the AlgaeTracker purchase. When you purchase one of those, you actually buy the communication plan from the company that sells them as part of the
Yes.
package. And they
Yeah, and
host
there,
all the data on their web server, so you don't have to worry about it. And it's available from their servers directly. And the communication takes place in real time to their servers, I think in the US, if I'm not mistaken, Colin.
yeah, Colorado, if memory serves, and they just
Yeah,
store all the different types of data, and you can download it at different points
yeah.
into like an Excel file, for instance, and then
Mhm.
plug away at it. But that's generally we're looking this upcoming year to putting that dashboard that they have
Yeah,
on our website, so people can check
yeah,
our website and see the same real-time data that we're seeing
yeah,
in our offices. So they'll start to see the little trends increase in phycocyan and be like, uh-huh, maybe there's a bloom coming,
yeah,
right? Which would encourage them to, watch our Facebook page, where we will put out, hey, we're seeing increases on our AlgaeTracker buoy. Be on the lookout for cyanobacteria. Looking for them too.
hmm. Yeah, we actually had the one for Grand Lake, the algae tracker for Grand Lake, on our website this past year. It's quite easy to pop that in there as an
We had a
item.
terrible time trying to get it to do it,
Well,
so
I
I'll
can
be
help you with
talking to
that part,
Brett.
maybe.
So
With my
yeah,
background in computing, so maybe I could help you with that point. Yeah. Okay, so another question. Are citizen scientists or volunteers being trained to use the test kits?
I would love to do that. We haven't done that so much to this point.
Yeah,
It's generally been myself and staff. But it would be really interesting to have some citizen scientists or volunteers in and around the watershed that could indeed assist with sample collection and testing. Um, Shockingly, I don't love driving up there every weekend to get cyanobacteria samples in the summer. So, because all major blooms occur on weekends.
yeah, that's
So,
hard.
I don't mind it, but it would be nice if there was somebody up in the watershed that could grab that sample and test it
Yeah,
on the weekend,
yeah, even in November. Okay, so
even in November.
yeah, we have a question from Cecilio Valadez Cano. Have you done molecular/morphological analysis to identify the taxonomy of the bloom-forming cyanobacteria? The taxonomy.
No, not yet. I haven't had the funding to do that kind of analysis, but I am hoping that this summer that will become available to us. There's some information that's recently come my way that we may be able to get the opportunity to actually test and see what species of of cyanobacteria it is. We haven't done it to date. I'm really keen to do it. I'm really keen to link it to the cyanotoxin testing. Yeah, excited about the possibility of finally getting to do some of that that testing. I'm really hoping that some of the agreements we have will come through, and that will be in the summer of 2024's future.
Okay, that's exciting. That'll be interesting to see if we can do that. Barb Glanville asks, I know Barb is from Skiff Lake out in the western part of the province. Did the test kits also have a shelf life? I think you said they do have a time.
They do indeed. Yes, Barb, they do. And it's like 6 months to a year. I should have brought one of the old test kits from the past summer up, because it has it right on the box.
Okay.
And but I know they do indeed have a shelf life. They do not last forever. So yeah.
Okay. So Pam Fowler says, thanks so much. Collected some cyanobacteria this fall, put it in a fridge. Couldn't find anyone to test it. Well, that's unfortunate. I think, Pam, you're from the Grand Lake area. So the process we're generally following is to send an email to gemseggrandlakewatershed@gmail.com on our website, or one of our board members, and they will try to get it tested for you if we have test kits available, which we did this last year in 2023. So, but that boils down to this outreach and volunteer training and having a coordinated effort among the watershed team, who has some resources to do this. Certainly, we have a lot of volunteers working, but not any staff at the moment. Hopefully, that'll change in the future. But that would be, a general marshaling point, one place where you can ask, can someone come and test it? How do you do that in the Belleisle Watershed? They just call you?
How do I do which?
How do you get a sample tested if someone's found one and collected it and put it in their fridge?
Either I myself go and pick it up,
Okay.
And you get our cyanobacteria information package,
Okay.
or one of the staff will pick it up if I'm not available. I try to do all the cyanobacteria calls myself if I can, because it lets me get out and talk to different community members and give them the rundown and skinny and all of this. So I try to get out to all the cyanobacteria calls that I can.
How does the general public know how to contact you? Is it on your website or?
Yeah, it is through our website and our Facebook. And I will tell them to email us or message us on Facebook. Or in most instances, I will put up my cell phone number and they can call me directly.
Okay, that's great. Yeah. So I guess for the Jimseg Grand Lake Watershed, Pam, we're trying, but we're not quite to the level of the Belleisle Watershed at this point. We don't have a Colin to kind of hop in his tank and come visit us. So someone says prevention is the best medicine.
I agree.
As prevention of nutrients in the water would
Bingo.
Be.
Yeah.
Has there been any effort this is from Lori Brown, has there been any effort to collect data on how much fertilizer is sold annually to farms in the catchment area?
Excellent question, Lori, but to the best of my knowledge, no, none of the watershed groups have looked into how much fertilizer is being picked up by farms. Fertilizers by farms are only one component of nutrient input. We would also want to try and find calculations for forestry operations. When you clear cut and those trees and forests are gone, you're going to increase nutrients. So you're going to want to look into what the runoff is by, hectare of cleared land. there would be calculations for nutrient input from septic systems that would add to it. But so there's a number of different inputs of nitrogen and phosphorus. There's of course natural ones. There's a lot of wildlife and stuff out there, and they're all going to the bathroom at some point. And they don't have the convenience of lavish modern bathrooms. So it's going into a lot of, I guess what I'm saying, there's a lot of crap floating around out there. So is the cruder way to put it. But no, that would be very interesting to collect data on fertilizers sold in the catchment area, and just see how much of that fertilizer is there. One of the issues that I see around Belleisle is when that fertilizer is put on. And I can't tell you how many times, and I've actually seen this because I drive through part of the Hammond River Watershed to and from Belleisle, and I have seen some farmers there around Darlings Lake just a spraying fertilizer the day before a great big rainstorm as fast as they can. And I want to get out and talk to them and say, what are you doing? Right? Because when you're spraying fertilizer on your field the day before big rain, you're losing a lot of money. Right? Like, guys, I can save you a chunk of cash and effort by just, giving up a little bit of field to help with riparian and just modifying when fertilizer is being put on.
Okay.
Right? You'd be bang, because then it has time to seep into the ground. And I see that in a number of instances where it's like gung-ho spray, and I know it's tough for the agricultural sector in New Brunswick, and I want to help them in any way that I can, but I do sometimes see, and I kind of scratch my head on all of it.
Yeah. Well, that's more of an education thing as well. So there's another opportunity there, I guess, for watershed groups when they have a few moments to do that. I have a question here from Stephen Smith. I often see very light green, tapioca-like globules suspended in the water in the Wolastoq. Is this a bloom or something else?
It well could be algae, and it well could be a bloom. It will it will occur throughout the water column. So you will get little flecks or globs of cyanobacteria in the water column. So without actually seeing it, I can't guarantee that's what it is, but I would have a sneaking suspicion that that's probably some kind of cyanobacteria or mat material that's occurring there, and is suspended in the water column.
Okay. Okay, there's a final question that looks like from Charlotte Valley, who's asking, has there been research also on the bloom's impact on wildlife? I know we had foxes at our property that disappeared about the same time as the dog died near the Bucket Club a few years ago. And
yes, cyanobacteria.
Is there any impact on waterfowl? I guess that's definitely related to wildlife.
Yes, it's toxic to them.
Oh,
it's toxic to a lot of other life. It's toxic to livestock. It would be toxic to foxes. They're in the they're certainly in the canine family, and they too would have scent. Cyanobacteria mats produce a scent that dogs are attracted to, that canines are attracted to. So yeah, they might eat it. When you have foxes around, you may also have some other issues related to people who may attempt to trap or shoot those foxes, which may explain their disappearance.
Hmm.
But certainly, yeah, they could drink the water or eat some of the bacterial matting and get sick and die. Um,
So it's the
and
same as
certainly it can cause
Go ahead, sorry.
fish kills, and it can be toxic to bird species.
Okay.
And it doesn't take much toxin. I mean, a really good amount of like a really about as much as my thumbnail is all the anatoxin A you need to kill a human.
Okay, not much.
Right? They are some of the most toxic substances on the planet when they're concentrated.
Okay. Well, that appears to be the end of the questions. I want to thank you very much, Colin, again, for sharing your broad and deep knowledge of cyanotoxin testing, and it looks like you'll be gaining more knowledge as we move down the trail.
I hope so. Every day is a school day. So,
Yes, exactly. That's
thank
part of
you.
part of the fun of life is doing research on new things.
It is. Thank you to everybody who attended, and again, a big thank you to the GemSeg Grand Lake and Brad for giving me this this, for lack of a better word, soapbox. So to pitch, and yeah, thank you to the Environmental Trust Fund who funded it. And to my watershed partners, Sarah, Roxanne, if you're both still out there or out there at all, could not have done this without your support.
Yes.
So,
Okay, great. And best of luck in the coming season in the Belleisle Watershed.
indeed, you too.
Okay.
So I'm sure you'll be hearing from me. I have a tendency to pop up like the bad apple. So.
Yeah. Okay, Colin. Take care. I'll be sending you a list of the questions everybody asks from the chat session so you can see them firsthand.
Okie dokie. Thank you.
Okay. See you
Have
later.
a good evening, everybody.
Bye.
Bye.